Students, let's today talk about chapter 6, molecular basis of inheritance. By molecular basis, this means I'm talking about the DNA. Let's see how it was known that it is the DNA that acts as a genetic material. In fact, by 1926, the quest to find the genetic material reached its molecular level. In fact, previously everybody used to think that it is a protein, a better candidate for acting as a genetic material. But in 1928, something happened that was called the transforming principle given by Griffith that changed the views of the scientist. So let us see what exactly happened in 1928. So in 1928, there was a scientist called Frederick Griffith. He was a microbiologist and he was working with a bacteria called Streptococcus Pneumoniae. This bacteria is responsible for causing pneumonia in mammals. Now what he found was that this bacteria is consisting of two strains. One is the smooth strain and the other one is the rough strain. Smooth strain means it produced shiny colonies, whereas the rough one produced no such colonies. In fact, they were rough colonies, they were not shiny. And what else was more important, that this S type of bacteria was able to cause pneumonia. This was a virulent strain, whereas the R type was non-virulent. The S type, the shiny coat, was because of the mucus polysaccharide coat. So you need to learn all these things for boards. So what you need to learn is that it was the Griffith experiment. What was his object? He was basically a microbiologist working on this bacteria Streptococcus pneumoniae. It consisted of S and R strain, and the smooth colonies are because of the mucus. This is a polysaccharide coat, the shiny colonies, and this was virulent. Now what he did was, he was working with the bacteria and he prepared injections of the S strain, R strain and injected into mice, and the mice died. Whereas when the S strain was injected, the mice was live. Now Griffith was able to kill bacteria by heating them, and he prepared another injection which was having the heat killed S strain, and this was not able to kill the mice because by heating the S strain was destroyed. The bacteria was destroyed. Now when something else happened when he added this S strain heat killed with the live R strain and injected into the mice died. Now Griffith concluded that this R strain bacteria had somehow been transformed by the heat killed S bacteria, and there was some transforming principle. And that's how the name of the topic is, transforming principle. Transforming means that the R type of strain was converted or transformed into a S type because of some chemical, but he couldn't say what exactly was the chemical. The biochemical nature still remain unknown. Then came the other three set of scientists, Oswald Avery, Colin MacLeod and Maclean McCarthy. That was somewhere in 1933-44. Now they refined the same experiment of Griffith and did it in a better way. So what they did, they purified the biochemicals, protein, DNA and RNA from the heat killed S type S cells, to which they added the live R cells. And they discovered that DNA alone from the S bacteria caused R bacteria to be transformed. So they also discovered that the protein digestive enzymes, like suppose if they take proteases and RNases, see how it is written, RNases, did not affect transformation. So the transforming substance was not a protein or RNA, but it was only DNA. So digestion with DNase, and suppose if we add DNase into one of the test tube, so this was again inhibiting the transformation. So finally by 1933-44, by this experiment, it was concluded that it is the DNA that acts as a genetic material. But still some of the scientists, they were not impressed. So there was an unequivocal proof that DNA acts as genetic material came from experiments of Alfred Hershey and Martha Chase in 1952. So the final conclusion, if we say, suppose who gave the final conclusive experiment to prove that DNA is the genetic material, so it will be Hershey and Chase. They worked with a virus that infect bacteria and the bacteriophages. What Hershey and Chase did in 1952, they worked with the bacteriophage, that is T2-phage. T2-phage, is a virus, threadpool-like virus, which has tail fibers and DNA as genetic material. Now, by this time, it was known how this virus used to multiply. This virus infects the bacteria E. coli and it multiplies on the E. coli machinery. Now, there are only two things present in the virus. There could be a protein coat and a genetic material called DNA. Now, because the virus has only two things, it has sulfur as in the protein coat and has phosphorus in the DNA. So, whatever goes into the bacteria and makes more copies will be the genetic material. So, what the experiment was, let's see. So, what they did, Hershey and Chase, they in two sets of experiment, in one set they chose sulfur, radioactive sulfur S35, and they let it get incorporated into the protein cover. So, this red color. And in the other experiment, the radioactivity was incorporated in the DNA. And you know why they chose only sulfur and phosphorus? Sulfur because it is a component of protein, whereas phosphorus because it is a component of DNA and not the other way. So in one set, when the sulfur was incorporated in the protein coat, what they found was that the radioactivity was found in the supernatant, whereas in the other experiment where they had phosphorus incorporated with P32, the radioactivity was found within the bacteria, that is in the pallets, the heavy pallets. So the whole experiment happened in 3 steps. The first step is called infection, where the phage was made to infect the bacteria. The second step was blending. Blending means the ghost, whatever is the empty thing that is outside has to be removed from the bacterial cell. And the third step is called the centrifugation, that these two things have to be separated on the basis of density. So from this experiment, finally it was concluded that it is the radioactivity when it was found in the DNA within this bacteria, and it is the DNA that acts as a genetic material and not the protein. Now, although by Hershey and Chase experiment it was proved that it is DNA acting as a genetic material, but then there were some viruses they were discovered which were having RNA as genetic material. Viruses like TMV and QB-phage, bacteriophage. So exactly what should be the properties of DNA that it is acting as a genetic material? Let's see. So the genetic material should be able to replicate. It has to undergo replication. It should be chemically and structurally be stable. And it should be able to provide the scope for new changes, that is mutations, although at a slower rate. And it should be able to express itself in the form of Mendelian characters. So if these properties are satisfied by whatever may be the genetic material like DNA or RNA or even protein, so that will be the better genetic. Let's see whether the DNA and RNA both are able to satisfy these kind of properties. So for replication, yes, both DNA and RNA, they are able to duplicate and they are able to make their replicas. But proteins they cannot. So protein we can minus out that the protein, they are the genetic material. Now the second property was stability. Now DNA has a sugar of this type, where at the second carbon there is only H, there is no hydroxyl group. So that is why it is deoxyribose. Now this makes it more stable, whereas in RNA it has a OH group here and that makes it less stable. So DNA is more stable than RNA. Also DNA has thymine, whereas RNA has a uracil group, which is again making it less stable. So DNA is better stable and therefore it is again a better candidate. Let's see if there is a scope for mutations. So yes, there is scope for mutation in DNA as well as in RNA. But because RNA is less stable and it is labile to temperature also, it is able to degrade faster, and it is also it also acts as a catalyst. So hence it is more reactive. So DNA is chemically more stable and less reactive and structurally stable, thus as comparison to RNA, and thus it is a better candidate. Although in some of the viruses we do have RNA as a genetic material, but proteins as such are out of the race for the genetic material. So finally it was proved that it is DNA which acts as a genetic material. So let's go and let us see what is the structure of DNA. So DNA is basically a helically coiled double stranded structure, like something like this. It has two ribbons with the base pairs at 90°. Base pair means I'm talking about two bases. Adenine suppose bonds with thymine and guanine bonds with the cytosine. And at the, if we try to see the molecular level, it is something like this. There will be sugars and phosphorus, and at 90° there will be bases. When I say it is a base pair, I'm talking about two bases together attached to the skeletal structure, to the ribbons, and the bases are always attached at carbon number 1, and the strand in front will always be having a other direction. Now suppose this is the sugar, the main component. It has a, at the third carbon it has a phosphorus, and at the first carbon it has a base. This base bonds with the other base of the opposite strand, and this will also be attached at carbon number 1, and thus the two strands will always be running anti-parallel. Anti-parallel means if this is the fifth carbon here, so this carbon will be bonding with phosphorus at the third 3 dash, and in the opposite one it will be the third dash on the upper side and 5 dash on the lower side, because they are running anti-parallel. This is how I made this oxygen on this side and this. So the two strands, they run anti-parallel, and you need to learn this structure, how they are paired. So a DNA structure looks something like this. It looks like a ladder with two strands and the stairs at this way. Now this is what we call as the base pair. Let's see what are the components of a genetic material, your DNA. DNA is made up of sugar, phosphoric acid and bases. Sugar is the deoxyribose sugar, it's a pentose sugar, and the structure is like this. At the first carbon there will be always a base attached here. Then there is a no oxygen at the second carbon, that is what is deoxy, and there at the fifth carbon also there is a CH2OH group. Then the phosphorus, the P that we talk about is phosphoric acid, H3PO4. Then the bases are two types, purine or pyrimidine. Purines are double ringed structure. They have a hexagonal ring and a pentagonal ring. Whereas the pyrimidines are a single ringed structure. It's a hexagonal single ring. So purines are again of two types, adenine and guanine. And pyrimidines are of three types, cytosine, uracil and thymine. Uracil is only found in the RNA. Now when I say it is a nucleoside form, so it is basically I'm talking about sugar and base. Sugar means the deoxyribose sugar with the base, and the base has to be at the first carbon. So how to name the bases then? Suppose it is sugar and cytosine, so I'll call it as cytidine, or if it is DNA, I'll call it as deoxycytidine. Similarly the other bases and the sugar, the other nucleosides. If it is a nucleotide, so that time it is a phosphorus also getting added, and this adds at the first phosphorus adds at the fifth carbon. So there will be a sugar, deoxyribose sugar if it is DNA, a base at the carbon number 1, and as phosphorus at the fifth carbon. And the names now will be cytidilic acid, thymidilic acid, adenylic acid, or we can say it is a phosphate, guanosine phosphate, because the phosphate group has been added, and this phosphate group makes it acidic. So let's see what is the structure of DNA as given by Watson and Crick. It was in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helical structure. This is Rosalind Franklin with her X-ray diffraction method. So when X-rays were passed through DNA, a structure something like this was obtained. Another scientist whose finding affected the model of the structure of the DNA was Erwin Chergaff. He gave certain rules. What he said that the total amount of purine and nucleotides always equals to the total amount of pyrimidine. That is A plus G, the total amount is equal to T and C. So the proportion of A is equal to the proportion of A is equal to T and of that of G equal to C. But because the two things are total, so A is equal to T and G should be equal to C. So it was in 1953 that Morris Wilkins, Rosalind Franklin took X-ray diffraction pictures of crystalline model and concluded that it is a long molecule consisting of two similar strands running in parallel and helical manner. They also concluded that the structure also showed regular stacks and the helix makes one complete spiral turn every 3.4 nanometer and has a diameter of 2 nanometer. The structure shows adjacent stacks of 0.34 nanometer. But it was Watson and Crick who worked on these clue of both Rosalind Franklin, Wilkins and Chergaff, and they proposed this helically coiled model. Let's see how this model looks like. Watson and Crick's model looks something like this, that the two DNA strands, they are helically coiled, and this coiling is producing two grooves, like major groove, then there is a minor groove, and this one coil, one helix is 3.4 nanometer and the two base pairs are at a distance of 0.34 nanometer. And the distance between the two strands is 2 nanometer, that is 20 angstroms. How the bases are attached, it's something like this. Bases. Let's see the salient features of double helical structure of DNA. So the first thing is that it is made up of two polynucleotide chain where the backbone is constituted of sugar and phosphate and the bases project inside, and the two chains, they are running anti-parallel. If one is having the polarity from 5' to 3', other is having 3' to 5'. The bases in the two strands are paired through hydrogen bonds, forming base pairs. Adenine forms 2 hydrogen bond with thymine from the opposite strand and vice versa. Guanine is bonded with cytosine with 3 hydrogen bond, and as a result always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of helix. Then the two chains are coiled at right-handed fashion. The pitch of the helix is 3.4 nanometer. Pitch means I'm talking about the one helix. So I'm talking about this to this. The pitch of helix is 3.4 nanometer, 10 to the power minus 9 meter is 1 nanometer, and there are roughly 10 base pairs in each helix. The distance between one base pair to another is 0.34 nanometer. The plane of one base stacks over the other double helix, and this in addition to hydrogen bond confers stability to the helical structure.